All right. So this part is all vocabulary. The main purpose of this lab was to teach you vocabulary, right? The first thing we talk about is the period gene, the promoter, the regular, and the reporter. So in your lab, right, they talked about these for part one, or before you did part one. So actually, I'm not going to rotate here. So what's the promoter? The promoter is the thing that it tells a specific injected segment of DNA to begin replication, right? The promoter is a regulatory thing. What it does is it tells a specific segment of DNA to begin transcription and become RNA. That RNA will then hit a ribosome once it leaves the nucleus and produce a specific protein. Now, why do we care about all this? Because we're trying to make GMOs, right? This is how you make GMOs. You inject certain things with DNA and you tell them to replicate themselves. And it doesn't always work, and we'll get into that later. But all of these injected artificial genes have a promoter. It's the regulatory region. It tells this specific gene when it needs to activate, when it needs to be transcribed, when it needs to be replicated. So let me write that down so that you'll have something. So your promoter is your regulatory gene. Your on-off switch. Now, your reporter, it reports what the promoter is doing. The reporter reads how much it's replicated. Or transcribed. So, why is the reporter necessary? Because, as with all things, all organisms must maintain homeostasis, right? We're trying to stay balanced. And in order to stay balanced, the reporter must be there. The reporter's job is to make sure you're not overproducing a specific enzyme or underproducing a specific enzyme in order to, or over or underproducing a specific gene to express a certain trait. In the lab, the experiment focused on genetically altering flies. And the only way you could tell if a fly was genetically altered was with red or white eyes, right? If the fly had red eyes, your genetic alterations worked, aka your experiment was a success, and you were able to get flies from being red-eyed or from being white-eyed into red-eyed. Now, how did those eyes turn red? Because the DNA you injected into them got red, got replicated, and was told by a promoter to begin transcription. The RNA made through that transcription process went to a ribosome. That ribosome made a protein. That protein became an expressed gene, aka red eyes. These two things work mainly so that you can get the specific traits that you're looking for, and the reporter makes sure that your organism doesn't die in the process. So let's get more notes now. Sorry, I gotta scroll through all of this. So the next thing we need to talk about is the period gene. Now, when we're talking about the period gene, the thing you want to think about is time. How often a specific gene is replicated depends on the period gene. The period gene is what tells your promoter to begin producing at a specific rate, and it will control that, right? It's what turns the promoter on and what it turns it off, right? The reporter will also communicate to the period gene whether or not it needs to turn off. But that's basically what it does. It controls the first two. Now back to notes. Sorry, I'm like tabbing between notes from class and trying to get these down so that y'all actually understand this. Anybody confused on the first three terms and why we need them? Just a quick no, and I'll continue. All right, let's keep going. So the next part talks about construct DNA. What is construct DNA? Construct DNA is constructed DNA. It's DNA you made in a lab, right? It's what is going to be injected. So this is going to, we'll call it the pre-made DNA. Now this pre-made DNA is what is going to be injected into whatever you're trying to modify, right? Your flies in the lab got injected with construct DNA, and this DNA is what attached to the regular DNA for the flies and replicated itself, right? Once it got replicated, it was able to be read and expressed as a gene, and blim, 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 blam, your flies have red eyes. I don't know what I did there. So these construct DNAs have, these construct DNAs have promoters and reporters, right? They attach themselves to the period gene of a fly, but they themselves have promoters and reporters. So here's what a construct DNA would look like, right? You have your, oh wait, let me do this in different colors. So we'll call this, we'll do it in blocks. We'll call this the what you want expressed, right? So we'll call it expressed. Now, if I want this specific segment of DNA expressed, I need a promoter and a reporter. So the promoter goes here. Let me go back to my notes real quick. Whoops, sorry, messed it up. Sorry, the reporter goes there first. And before that is your promoter. Yeah, I was wondering why it looked weird. Now, why is your promoter not connected to the main one? Simple. The reporter tells it when to turn off. Best not to put your activation thing right next to the specific gene in case it doesn't work properly, right? So this is what it would look like, and it would be on a bit of a loop. Oh, whoops. Here. Sorry, I got to do that. And it would look like that. Now, what is this loop made of? It's made of extra DNA. That's it. It's just made of extra DNA. But these two triangles at the end, now you might be confused as to what those are. Those two triangles at the end help our construct DNA attach to the to the fly's original DNA. These two ends will construct or will make contact with the fly's DNA and incorporate them. And so what you'll have is a bunch of other DNA on the end. Once it enters the fly, it will connect itself. And here you'll just have one long strand of nice continuous DNA, minus the two triangle bits. Now those two triangle bits are made of more bases to allow them to connect. Now, what happened to that giant loop string part of extra DNA? It got disconnected. Its only job was to hold the construct DNA together, because the construct DNA can't hold itself like that. Not until it's connected, right? The two ends, while yes, they do maintain its shape and allow it to connect to DNA, it can't stay together without all that extra DNA looping around. Now, that might not be in the test, but it's just good to know. So that's your construct DNA. Now back to my notes. Cool. Oh, hang on. All right. Now, the next thing. I gotta scroll through these. Hang on. Because a lot of this is just more of the lab that wasn't necessary. All right. Cool. So okay, I'm just gonna talk about the lab from this point on. You've gotten most of the vocabulary that you need to understand. So from this point on, we're just gonna talk about the lab. So this is the end of part 1. Beginning on part 2, the experiment actually starts. Now that you have construct DNA, what you're going to do is you're going to take all of it and you're going to inject it into some embryos, right? When you inject it into embryos, there's about a 10% chance that any of the genetically modified embryos will actually make it out. Why? Lots of reasons. One, you could have injected things wrong. You could have accidentally used you accidentally could have used embryos that were too young. You could have accidentally injected too much fluid, causing them to rupture. You could have accidentally missed the spot that you were supposed to inject the embryos with. So you instead injected them in the head, causing them to suffocate. Lots of very morbid things. But if the DNA gets in there and it does get incorporated like this construct DNA has to, there is another piece of DNA. Now, for the sake of this, we'll call it transformed DNA because that's actually what it's called. It's called transformed DNA. It's transform or transfer, I don't remember. But it starts with a T. This DNA, what it does is when it enters the embryo, this is what tells this is what gives the information for, or sorry, my brain is like completely farting on this. I'm sorry, guys. The transformed DNA's job is to be read and to be transcribed. This in and of itself is just DNA, but it has a very specific set of instructions. When it is read, it will make an RNA string that makes a very specific enzyme that attaches to the original DNA and attaches the construct DNA to it. The purpose of the transformed DNA is to attach the construct DNA. It has the instructions to make a protein that attaches the construct DNA. I don't know why that was so hard to say, but that's what it does, and you should know that it exists. And I don't know why the A looks so fancy there. Now, once all that is injected, you simply wait for the flies to grow up. They do their thing, and you see if they've actually got the trait. Now, this is probably going to be on the lab. It is not guaranteed that you will always get the results that you're trying to get. Sometimes all your embryos died and you weren't able to make any proper adult genetically modified flies. Sometimes they all just die prematurely. Sometimes they're infertile and they can't make babies. Sometimes you accidentally killed the embryos by injecting them with too much fluid. I did that on this lab. All my flies died. It was sad. Yes, that is also entirely possible. Rerun the lab if you want to. It is possible to screw it up pretty badly. Now, since it is possible, always account for the fact that you can screw it up the first time and reread the experiment to see if you did everything correctly, right? Other than that, that is all from the labs. All of this is just basic genetic information. Now, there are also 2 types of cloning that you need to go over, but those are in the notes, and the notes are long and confusing, and I don't understand them. So I'm sorry, I can't help y'all there. Unless I ran through them right now. Give me 1 second. Come on, where is it? Sorry, I gotta pull them up. All right, hang on. All right. Yeah, other than that, I think that's it. So for, hang on, I'm just running through this really fast. All right, so let's talk about cloning for a second, and then I'll disappear while I post this on YouTube. So for cloning, what you really need to know is there's two different types of cloning. There's the kind in which you use an egg embryo, and then there the kind where you use normal diploid cells. If you use, they're differentiated mainly just by the cells that they start with. But all right, hang on, sorry. But in theory, you can take the DNA from one animal or organism, inject it into an egg of another animal, fuse them, and cause the and then take that, we call it a fertilized egg, right? Take that fertilized egg that you injected with DNA, put it into a surrogate, and it can have uh, a a clone, right? Now, how does that work? Basically, what you do is you take the DNA from one animal. Here, you know what? Let me just screen share. Basically, you would take the DNA from sheep A here, take the egg cell from sheep B, remove the nucleus with all the DNA in it, put the DNA in that previous thing without a nucleus, and then fuse them, slap them in a surrogate, and then bam, you got a clone of sheep A. Now, how does that make sense? I don't know. I wish you would have taught us something. But yeah, biggest example of a cloned animal is Dolly the sheep. If y'all have never heard of her, look her up. Dolly the sheep is basically a cloned sheep. Now, granted, it didn't live for that long because you can't just clone an animal without consequences. But yeah, that's basically how that works. Other things that we need to look at are, hang on. There's another thing here that I wanted to look at. Plasmids. What are plasmids? Plasmids are circular DNA. Remember that circle of construct DNA that I drew for you? It's technically a plasmid. And that's it. Yeah, that's it. All right. Hopefully y'all learned something today. Any questions? Anybody? Please, this test is going to be really difficult. No? Nobody? Nobody want to ask anything? Oh, I should probably stop the recording.